Today we're going to talk about the urinary system. We will talk about evolution of the nephron, a little bit about histologic features of the nephron, the kidney, and other components of the urinary system, and how each of these components contribute to unique functions of ridding the body of its waste and preserving homeostasis of the body. Enjoy. The objective of the urinary system presentation is to look at the anatomy. We want to see the structure of the kidney, the different components of nephrons, identify the nephron components, as well as excretory components, and look at the function of the urinary system. Here we see the glomerulus here and here and here, and the parietal epithelium that lines the outside of that as well. Inside there we see the visceral epithelium, and we see that here these are capillary cells into there. We can see fenestra. Here we see the cortex and medulla of the kidney. Urinary system, it functions to rid the body of waste, urea, uric acid, creatinine, and salts, and it preserves the constancy of extracellular fluid in composition, volume, and pH. In other words, it helps maintain homeostasis. It also has an endocrine function. It secretes erythropoietin, which is a protein that stimulates red blood cell production. It also secretes renin, which interacts with blood proteins to ultimately cause release of aldosterone from the adrenal. So it has an endocrine function, makes blood vessels, makes blood cells, and also it releases aldosterone. So in the urinary system we can see the two kidneys on either side, major blood vessels going into the artery and veins going in a little hilus region of the kidney, and then we see ureters coming down from each kidney down into the urinary bladder, and then ultimately fluid would come out the urethra. So here we can see the kidney. This is the cortex. There'll be a capsule around that cortex and medulla. You can see the medullary pyramid, shaped like a pyramid, in through there. So this would be the cortex, and then in through here would be the medulla, right in through there, the adrenal sitting right on top of the kidneys. You see the sinus in through there. We got a minor calyx and a major calyx, which ultimately goes to the renal pelvis, that goes to the urethra, and from there it goes to the urinary bladder, and then in the case of a male, it will come through the prostate, and the urethra will come through the prostate. So we got major arteries and veins going to the kidney, and here we see the nephron. The nephron is a functional and the basic units of the kidney, where you have blood vessels that interact with the nephron at the glomerulus, and so you have a first capillary network of a portal system, and then the efferent arteriole is actually a portal arteriole, and then you have the peritubular capillaries is the second network of the system. So we'll go through these different components. Cortex, and you see how the nephron fits right in there, so that your collecting tubule goes down through here, but also the long loop of Henle extends down into the areas in certain of the nephrons. So this would be major calyx, minor calyx around each of the medullary pyramids, draining it. Fluid goes through here to ultimately to go to the urinary bladder. So we have the cortex, the capsule that surrounds the kidney. We have the medulla, this portion right in through there, and from there we'll have transitional epithelium that goes off of its surface. Here we see a glomerulus, the parietal epithelium, and the visceral epithelium located, which actually surrounds the capillary tuft. Here we see the arcuate arteries. These are the name of it. Looks like it makes an arc, and from that you have the blood vessels that run through the glomerulus would be coming off of these arcuate arteries that will be in here. So they go right between the medulla and the cortex area. We can see these arcuate arteries. If we look more closely at the nephron, we see it starts with the glomerulus. And so we have a capsule that's around a glomerulus. This is the parietal epithelium, and that parietal epithelium is continuous with the proximal convoluted tubule. Proximal convoluted tubule becomes a convoluted portion and a straight portion. It goes down to the thin portion of the loop of Henle, which then comes back as a thick portion of the loop of Henle. Now it's a distal tubule, and that will actually come back up and make contact right in this region of the vascular pole versus urinary pole. The vascular pole of the nephron, and then finally the fluid goes through a collecting duct that goes in through here. So in the capsule, we have dense irregular connective tissue like all other capsules, and below that we have the cortex. And in the cortex you have mostly proximal tubules, but you also have distal tubules as well. Some loop of Henle, but not that much. And we see a lot of glomeruli located in the cortex. Here's the glomerulus. You can see the parietal epithelium in through there. Visceral epithelium covering each of the blood vessels that are located in through here. These are proximal tubules they have a brush border. Also the nuclei far apart. Look how far apart the nuclei are on these cells as opposed to this. This is a distal tubule. See, the nuclei is much closer than these guys. Look how far apart the nuclei are. Also, this has a brush border. This does not have the distal tubule does not have a brush border. So the parietal epithelium is continuous with the proximal tubule. And here we can see where the parietal epithelium surrounding the capsule. This is the renal corpuscle. It starts the urinary pole, where this is filtrate that will start the urine. Also located right in through here in a distal tubule that comes back up there, you have a high density of nuclei located right there. We can see those right there in the distal tubule. Proximal tubule with the brush border, which you see is PAS staining. No brush border in the distal tubule. This is the macula densa. Macula densa is an osmotic sensor. It senses the osmotic pressure in there, and it can cause the release of renin, which is located in the afferent arteriole that we can see. So here we can see this is the urinary pole, the vascular pole, where the blood vessels go in and out. So you have the efferent arteriole right in through there with a capillary tuft in through here. Here we can see the macula densa and arteries going into the capillaries tuft, and we see the red blood cells in through there. This is one of those capillary tufts, so this would be the visceral epithelium, the vascular pole. Here's the urinary pole of this, and that we can see. These tubules here are proximal tubules, some distal tubule right in through there, but these with this flocculent stuff is really the brush border as opposed to simple microvilli of the distal tubule. Again, we can see the glomerulus with the parietal epithelium in through there, the urinary pole. If we look at an electron microscopic view, you can see the podocytes, which are the visceral cells that actually locate to, they touch the urine. They also touch the endothelial cells. This is the endothelial cell that line the capillary, and we can see the fenestra. These are the cross sections of the fenestra, and we can see the fenestrated epithelium there. The podocytes also touch these endothelial cells, and you can see for their little foot-like processes have spaces in their filtration slits where fluid goes, blood fluid comes from here, goes in through this space, and once it's in this space, Bowman space, it would be filtrate, or the beginning of urine. Here we see a couple glomeruli. This is one of the vascular pole, another vascular pole, and this is the distal tubule that's right up next to it. These are distal tubules right in here next to this one, and there is a urinary pole as well. This is just a portion of a glomerulus. If you cut one straight through here, you'll get some of these blood vessels in the glomerulus. This is a protoepithelium. For sure, these are proximal tubules in through there with the brush border, and some distal tubules can also be found. Now, located in the afferent mostly, and also a little bit efferent arteriole going out through there, are these renin granules, and you can see these granules here. They are PAS positive. So the PAS is allowing us to see the basal lamina of these. Actually, what we see in there, the blue there is the basal lamina, also the brush border, but it also lets us see in the blood vessels these renin granules, which are released based on the osmotic detection of the macula densa located right in there. It should be hypotonic. The urine should be hypotonic. If it's not hypotonic, it will cause renin to be released, and ultimately aldosterone be released to increase sodium absorption. Now, there are a couple of things in the cortex. There are these cortical nephrons, which are located even with this thin little loop of Henle up in the cortex. In contrast, there's juxtamedullary next to the medulla nephrons with a long loop of Henle. And so, regardless of which type of nephron, the cortical nephron or the juxtamedullary nephron, the fluid goes into the collecting tubule, and the collecting tubule goes down through the medulla. So in the medulla, you'll have the collecting tubule of all the nephrons. You will also have the long loop of Henle located in through there as well. Proximal tubule is right in through there, distal tubule right in through there, and we can see the renin granules that will be released. Here's in the cortex. We're right up here in the cortex. This is the beginning of the proximal tubule. So this is the urinary pole, and this is the vascular pole, and you can see where blood vessels are coming into that region. So if you look right in through there where the proximal tubule is thick portion, is going to give rise to the thin portion of a loop of Henle. And we can see that transition right here. This is brush border here, characteristic of the proximal tubule, and all of a sudden it becomes thin. There's another one right there. Okay, so this is a transition between these two, thick portion and thin. Thick, thin, thick, thin, that we can see, and that's what we're seeing right here. So this is the proximal tubule. Here's the distal tubule. The distal tubule doesn't have a brush border, but it has lots of mitochondria that we can see here. So this is distal tubule. So what we're seeing here is a transition between these two right there, and then we see various profiles of the thin portion of the loop of Henle, and then we see the thick portions of the distal tubule coming back up into there. So that's what we're seeing. So in a medulla, you will see collecting tubules, which are known by columnar, could be columnar cells, and distinct boundary between adjacent cells. We also see proximal tubules with the brush border and also a big distance between adjacent cells. We should be able to see distal tubules too. I can't say if I see any distal tubule. Maybe this is one in through here. It certainly is easier to see. If this is distal tubule, then that may be another transition from loop of Henle to the. But you don't really see that much of the thin portion of loop of Henle in the cortex, but you do in the medulla. So that's what we're seeing here, is a thin portion of the loop of Henle. So again, we can see in the medulla, we can see collecting tubules, blood vessels in through there. We can see thin portion of loop of Henle, and we can see the distal tubule going in through there. Here again, we are down here. If you look at this one, we are located down about midway in the medulla. And here's the collecting tubule with a distinct boundary between adjacent cell, another one right here. We see blood vessels that run through there. And we see the thin wall, really thin wall of blood vessels that are running through here, as opposed to a little thicker wall of the thin portion of the loop of Henle. So this would be the thin portion of the loop of Henle right here and here. And I'm not sure whether or not this is the ascending portion of the thin loop, because the descending portion of the thin loop has large nuclei located in there. If you look at the cytoplasm of this guy, it looks different from that. And this and that are descending, and that is descending portion. This is the blood vessel right in through there. You can see the thickness between the endothelial cell, there, real thin, and the loop of Henle, the thinness of the loop of Henle. And so we can see here, this is a thick portion of the loop of Henle and the thin portion of loop of Henle in the capillaries. And the loop of Henle, the nuclei, projects out into the lumen very abruptly, much more so than do the endothelial cells. So again, we can see that the ascending portion of the distal tubule, and we can see it doesn't have a brush border, no brush border, collecting tubules. Again, we're right down here in the medulla, and then we see the thin portion of the loop of Henle right in through there, the smooth profile, and also the rough profile of that one. And in between each one of these, you're going to see little capillaries in through there, so you have very close exchange between the capillaries and the nephrons. So here we see back in the cortex, we can see this is the renal corpuscle. But what I wanted to point out to you is that this little structure right here is a thin portion of the loop of Henle. And remember, with cortical nephrons does have a little bit of loop of Henle right here, not nearly as big, as elaborate as what we have in the juxtamedullary nephrons, but there is a little bit, and you can occasionally in the cortex find the loop of Henle, because the entire nephron is in the cortex, cortical nephron, as opposed to a juxtamedullary nephron. Here again, we can see the cortical nephron, and these two nephrons have a little different function. One is it removes a lot of fluid, and the other one, by having a pin loop, a loop like the hairpin loop, you're able to change the osmotic pressure in through there. So what we're seeing here is a heat source, and this is the fluid coming down. So the same amount of calories is going into this heat source in both of these. But if you have this hairpin loop like that, then you're able to increase the temperature within the contents with the same amount of heat that we have with a straight tube. So this is characteristic of the cortical nephron, and this is the juxtamedullary nephron, where in the case of a nephron, this is not heat, this would be osmolality is increasing, salt concentration is increasing as you go down, and that allows it to create a gradient down in through a high concentration of salt. So when the collecting tubule comes down through this high concentration of salt, it reabsorbs the fluid again. Remember, there's 180 liters of fluid that's reabsorbed each day, and it has to be a constant reabsorbing fluid, and it does by set up this gradient that pulls the water back out of the nephron. So here we see the kidney. This is in the cortex, and we can see the basilar rays running through there. There's the glomeruli running through there. But what I wanted to note is that the arcuate arteries. So we see a couple arteries right in through there, and the arteries right in through there. So right in through there is going to be the arcuate artery that goes in through there that feeds the nephrons. As we go down toward the end of the medulla, we'll pick up these simple columnar epithelium of the collecting tubule, but we also see blood vessels in through there. This is actually part of the vasa rectae, blood vessels that come in through there, and then we can see the loop of Henle as well. And so we can see that the afferent arteriole is coming in as very small. Actually, the afferent is larger than the efferent. It doesn't look that way here, but it is. There's the first capillary network, and then the second capillary network is around the tubules to reabsorb the fluid. So fluid comes in, goes in through there, and is reabsorbed immediately. That's how you reabsorb 180 liters of fluid today. Arcuate artery giving branches off that feed the glomeruli. Also, the arteries and veins go down here with the vasa rectae that goes there to provide oxygen and remove salt. So down these juxtamedullary nephrons project way down into the medulla, and that's the ones responsible for the concentration gradient. That's a higher concentration of salts are located in through. So when a collecting tubule goes there, it reabsorbs the fluid. So here we can see where the vessels were not perfused, and so we see the red profiles of the vasa rectae that goes in through there, which takes nourishment down in through there. Here again we can see the distal tubule, and here you can see how the nuclei really bulge out in the thin loop of Henle versus the capillaries in through there. Here again you can see the capillaries and red blood cells and how close it is to these 1, 2, 3 profiles of the loop of Henle. Here we see profiles, loop of Henle, collecting tubule, distal proximal tubule. And as you go on down here, ultimately you get transitional epithelium. And so when we look at the very end, this would be the minor calyx, and there would be a major calyx in through there. The minor calyx has transitional epithelium that we have there. From the minor calyx becomes the renal pelvis. Renal pelvis goes to the ureter. Ureter has different profiles, about 3 profiles of smooth muscle, the interlongitudinal circular muscle that we see there with transitional epithelium. Here we can see it's 262 transitional epithelium and lots of smooth muscle associated with it. It's very important to remove the fluid from the kidney, and the ureter does that. In a dog, if there's a problem, oftentimes it's the bladder that pops because the ureter has done its job to remove the fluid from the kidney. And then from there you get to the bladder, the urinary bladder, and again you have transitional epithelium on its surface, and then smooth muscle bundles in various profiles that will be associated with emptying of the bladder. So what we talked about today is the kidney, the cortex, medulla. The fluid is drained from the urethra down into the urinary bladder and down into the prostate of the capus of man. We showed here the different components of a nephron, and here is the urethra of the penis, which is the opening of the penis. You see there's cavernosus tissue in through here, which is responsible for filling up with blood and causing erection. There's also erectile tissue around the urethra too to hold it open during the erection. Uses transitional epithelium, but other types of epithelium can be seen there too. Could be stratified columnar as well. So from the urinary bladder, we see that the urethra opening in through there, and then finally out through the penis, and it was in the prostate. This is the prostate here, that the system becomes a urogenital system, because the sperm coming up from the testis through the ductus deferens are going to be mixed with the seminal vesicles fluid right in the prostate, and then that will go through the urethra as well. And I didn't show you, but the female also is a similar type thing. There's the vagina and there's urethra of the female, and the urinary bladder sitting right in front of the vagina. So that's what we will see. We want to look at some didactic a test question for the final exam for 2011. I don't have 2012. So we'll go through 5 of these. Which cells of the immune system may represent B lymphocytes? Memory cells? Yes, B lymphocytes can be memory cells. Plasma cells? No. B cells make plasma cells, but they're not plasma cells. Cytotoxic cells? No. Cytotoxic cells are T lymphocytes. The answer is A. Which skin structure function matches? Gland for which our class was named. That's a mammary gland. Nourishment of young? Yes. The answer is yes to that. Sweat gland thermoregulation? Yes. Membrane coating granules protect against UV light? No. They protect against desiccation. The answer is D. Okay. Which of the following items of mucus, characterized mucus in the respiratory system, contain proteins that present odor chemicals to receptors of olfactory epithelium? Yes. Trap and washes dust away? Yes. Toxifies gases? No. It detoxifies gases. So the answer is D. The pituitary has little effect on regulation of hormone secretion in which of the following? Adrenal medulla? No, has little effect. That's true. Chief cells in the parathyroid? True, little effect. The testis? A lot of effect. So the answer is D, is A and B. Calcium concentration in the blood are affected by calcitonin from the thyroid? Yes, it's from the parafollicular cells. Absorption of bone from osteoclasts? Yes, that elevates calcium concentrations. And then the parathyroid hormone from the thyroid gland. Parathyroid hormone comes from the parathyroid gland, not from a thyroid gland. So again, the answer is D.